Chapter 26 is also one of my favorite chapters in A&P 2. Well, I would have to say A&P 1 and 2. And probably for two reasons. One, it's all physiology, and of course, that's my passion. But also, it is highly integrative. We are going to take a look at mechanisms that will pull pieces together from both A&P 1 and A&P 2. We'll start by reminding you about those fluid compartments and what that fluid composition is. We'll talk about osmolarity, we'll talk about electrolyte balance, and that will take us to acid-base balance. We will look at how the pH of extracellular fluid is controlled by chemical buffer systems, your respiratory system, and your renal system. So we will be again integrating information, especially from chapter 22 and 25, in taking a look at pH balance. So let's get started. This slide should be a reminder of the three fluid compartments within your body. Remember that on average, our 70-kilogram man is going to have about 40 liters of water. Now, 1/3 of that will be extracellular fluid. And most of the extracellular fluid is interstitial fluid between the cells, with only about 3 liters of plasma being part of the extracellular fluid. By far, 2/3 of the total body water is inside your cells, intracellular fluid. Now, before we move on to talking about the different solutes that are found in the fluid within the body, I want to remind you of a couple of things. This is something that I show my A&P 1 class. Remember your different fluid compartments. What is it that separates the blood plasma from interstitial fluid? You remember? That's going to be the capillary wall. Capillary wall. Now, what is it that separates your interstitial fluid from the intracellular fluid? That is going to be the cell membrane. And, if you'll recall, both of these barriers between fluid compartments are permeable to water, which is why we can have water move from one compartment to another down an osmotic gradient. Just a little reminder of your basic cell membrane properties from A&P 1. Now let's follow up with this idea. So, if it's true that these barriers between these fluid compartments are permeable to water, then let's talk about what influences this. The movement of water across the cell membrane, the plasma membrane, is controlled by what? If you said osmotic pressure, that is correct. But the movement across the capillary wall is primarily controlled by hydrostatic pressure. Again, a reminder of things we already know. So let's look at a couple of scenarios here. There are going to be changes in fluid shifts when there are changes in different things. So if you have a change in extracellular fluid volume, you would see, if we have an increase in plasma, you would see a shift of fluid into the interstitial fluid, which means there would be an increase there. And you would move more water into the intracellular fluid because of proteins pulling it there. Osmolarity, again, if you have an increase in osmolarity in the interstitial fluid, water will be drawn from the cells. Remember our discussion about tonicity in AMP1. So let's talk about some solutes here. Taking a look at this chart, we see things we already know. Take a look at sodium and potassium. These measurements are in milliequivalents per liter. So obviously much more sodium outside cells than inside. And we already know much more potassium inside cells than outside. We do have calcium in the extracellular fluid and none to speak of inside cells. Magnesium, low outside of the cell, higher inside. Bicarbonate ions, this is going to be important for us to know at the end of this chapter. We see many more bicarbonate ions outside of the cell than inside. The same is true for chloride. Remember, where sodium goes, chloride goes. Phosphate is higher inside the cell, as are sulfates. Now here, let's remind ourselves of proteins. We know that there are such things as plasma proteins. So we see proteins inside the blood plasma. We know that there are proteins inside cells. And very few, if any, proteins in the interstitial fluid. We already know that from our blood chapter. I'm using this image from the other book. I think it's a little bit more dramatic than the one in your textbook that shows you, in essence, water balance in the body. In general, when we talk about law of mass balance, we are talking about the amount of a substance that is eliminated has to equal the amount of a substance taken in. Another way to look at it chemically is the amount of reactants has to balance with the amount of the products. So in terms of water, the first case is the best scenario. Intake has to equal outgo. So we will be looking at water gain. How do we gain water? Well, food and drink. Metabolically, when we have chemical reactions that are dehydration synthesis reactions, one of the products is water. So through metabolism, we can gain a little water. How about water loss? Insensible water loss is sweating and exhaling water vapor when you breathe out. Urine output is water loss, and feces has water in it. And if you notice, we have our intake and metabolic production. We subtract our output, and we have our mass balance of water. This is the image from your book. I guess I just like the fact that there's a human being in the previous image. Let's take a look at the regulation of water intake. It is primarily driven by the osmolarity of the extracellular fluid. It is the hypothalamus that has osmoreceptors that are responsive to an increase in osmolarity. They will activate the thirst center within the hypothalamus, and one will then have a sensation of being thirsty. Just taking a drink really helps with the sensation of being thirsty. More importantly, water will be absorbed from the GI tract because you drank it, and that will decrease extracellular fluid osmolarity and increase plasma volume. Increase plasma volume. That's related to blood pressure, isn't it? Exactly. And so we take a look at this side of the figure and see that a drop in plasma volume, and that is a significant dehydration there, would drop blood pressure, and that would trigger the renin-angiotensin-aldosterone mechanism that we have barely looked at in chapter 25. We will look at in more detail in just a minute. Angiotensin II is a product of this mechanism, and it also targets the hypothalamic thirst center. One of the things that I learned in my first career when I was an elite professional athlete riding horses, especially here in Texas, it was really easy to become dehydrated. For that reason, I didn't want to get to this point, and so I made sure that I drank water or a sports drink throughout the day, and that kept me from getting into water balance trouble. We know already that the kidneys function to conserve water volume. Kidneys can't restore lost volume. All they can do is conserve fluid and prevent further loss through the urine. And I really like this summation, big picture, of how the kidney does that. We know that GFR is going to influence fluid output. As that fluid flows through the kidney, there is obligatory reabsorption of water, but there's also regulated water reabsorption, which we'll look at in a minute. And by triggering the need to conserve volume, the urine is more concentrated. Here is a flow chart from your textbook that simplifies the idea of water being regulated in terms of the kidney conserving it. Here we see the influence on the posterior pituitary, which then would secrete ADH, targeting the collecting ducts of the nephron and increasing water reabsorption by inserting more aquaporins. Remember that it is ADH that targets the cells of the collecting duct. Also remember that it is the osmotic gradient that is developed through the nephron loop and the vasa recta that determines how much water will be reabsorbed in the presence of aquaporins. We have about 300 milliosmoles at the top of the medulla and 1200 at the bottom. If you do not understand the influence of the osmotic gradient and ADH on being able to conserve fluid, go back to chapter 25 and review that information. This is a nice flow chart from the other textbook that shows the control of vasopressin secretion. Take a look at it, see if it makes sense, and let's talk about it more in class. Now let's turn to sodium balance. Remember, we talked about sodium reabsorption within the nephron. 99.5% of all sodium that is filtered is going to be reabsorbed. Most of that in the proximal tubule. Remember, that is obligatory. Water is going to follow that and chloride passively. 25% of sodium reabsorption occurs in the loop of the nephron, and then the final balancing act occurs in the distal convoluted tubule and the collecting duct. This is regulated. It is facilitated. It is controlled by two hormones, aldosterone and AMP, atrial natriuretic peptide. You should be familiar with the mechanism of sodium reabsorption, but let's take a look at the hormonal control of sodium reabsorption in more detail. This flow chart demonstrates the renin-angiotensin-aldosterone system, or RAAS. Angiotensinogen is a precursor to angiotensin. It is constantly produced by the liver. Now, nothing will happen to it until blood pressure drops. If you'll remember, that triggers the granular cells to release renin. So now we have renin and angiotensin in the plasma. Renin acts as a catalyst to convert angiotensinogen into angiotensin 1. That form will remain in the plasma, and as it circulates through the blood vessels, there are blood vessels, we think primarily related to the lung, but not only the lung, that produce an enzyme called angiotensin converting enzyme, ACE. ACE acts as a catalyst to convert angiotensin 1 into angiotensin 2. Now, that is a very significant hormone. Let's take a look at its effects. Let's look now at what effect angiotensin 2 will have on various target organs. One of the things it does is target the arterioles, in particular the smooth muscle in the arterioles, causing vasoconstriction. Angiotensin 2 also targets the cardiovascular center in the medulla oblongata. That increases cardiac output by increasing heart rate and force of contraction. It also is going to trigger vasoconstriction. These two target organs and their effects ultimately lead to an increase in blood pressure. We know this part, and now we know something that can target it. On this side of this page, we see that angiotensin II, we already know, targets the hypothalamus, triggering an increase in thirst. The other thing that it will do is cause the hypothalamus to increase the release of ADH, or vasopressin. Now, the final step in the RAAS process is the targeting of angiotensin II on the adrenal cortex. This causes the adrenal cortex to secrete more aldosterone. Aldosterone targets cells in the distal convoluted tubule and a little in the collecting duct to increase sodium reabsorption. When we look at the effects on this side of the page, that is going to increase volume, blood volume, and subsequently maintain osmolarity. When this happens, blood pressure will return to normal. Another hormone that influences sodium reabsorption is atrial natriuretic peptide. I talked a little bit about this in chapter 16, as well as in the chapter on the heart and the blood vessels, when we talked about blood pressure. An increase in cardiac return to the atria increases the wall stretch, and when the atrial wall stretches, it releases natriuretic peptides. These have a variety of targets as well. It targets the hypothalamus, and that produces less vasopressin. It targets the kidney in two ways: the tubule, decreasing sodium reabsorption, and the afferent arteriole, dilating it, increasing GFR and decreasing renin. The decreased renin will have the effect of lowering aldosterone, but AMP also targets the adrenal cortex to lower aldosterone. And then we also see AMP target the medulla oblongata, and the overall sympathetic output is decreased. When we look at these first three, we see that there is an increase in sodium chloride and water excretion, which will lead to a decrease in blood volume. And in combination with these items over here, you have a decrease in blood pressure back to normal. I'm using this slide to help you gain a little perspective. So what happens if you eat salt? Well, you will have no change in volume if all you're doing is eating salt. For example, you go to the movie and you get salted popcorn, but you forget to have anything to drink. So in terms of your overall plasma volume, there'll be no change, but you will have an increase in osmolarity. We know that targets the thirst center, and the hypothalamus releases vasopressin. Vasopressin increases water reabsorption. Your thirst center increases water intake. Now, in this pathway, we know that ADH, vasopressin, causes the kidneys to conserve water. Water intake is going to, and water reabsorption is going to increase extracellular fluid volume. That will cause an increase in blood pressure. Well, what happens when this occurs? Well, the increase in extracellular fluid volume will then cause the kidneys to excrete salt and water. It's slower than this first response. And the cardiovascular center, the reflexes there, will lower blood pressure in response to the raised blood pressure. And so overall, even if you eat too much salt in a given 5 to 20 minute period, your osmolarity, volume, and blood pressure will return to normal. How does this rate relate to blood pressure if you are eating too much salt chronically? Well, too much salt chronically can cause retention of plasma volume and therefore higher blood pressure. And that's one reason why doctors recommend to people who have high blood pressure that they cut back on their salt intake. This is a simplified flow chart showing you the balance of sodium and potassium. We talked about this when we talked about the fact that the transporters that handle sodium in the tubules, in the distal convoluted tubules, also handle potassium. As sodium is conserved or reabsorbed, potassium will be secreted. They are both under the influence of aldosterone. This and the next slide are the big pictures. Why have we been talking about water balance and sodium balance so much? There is one answer. Mean arterial pressure has to be maintained. When MAP is maintained, there is adequate perfusion pressure, ensuring that the tissues receive the nutrients that they need and oxygen, and that the waste products and carbon dioxide are removed. Mean arterial pressure. That's the whole reason. So take a look at this flow chart. See if you can follow the pathways. We are going to talk about this more in class, but also take a look at the next slide as well. The other thing to remember is we already know there's short-term regulation of mean arterial pressure. That's done by the baroreceptor reflex, the heart, and the blood vessels. We also know that short-term we can shift fluids from one fluid compartment to another. Long-term regulation is what is going to be handled by the kidney under the influence of ADH and aldosterone. Again, this flow chart, instead of blood pressure, shows you what would happen under condition of dehydration. Well, if you're dehydrated, your blood volume and blood pressure are going to drop. So take a look at this mechanism and see if you can follow, but break it up into chunks. Look at the cardiovascular mechanism, renin, angiotensin, aldosterone system, the renal mechanisms, and then what the hypothalamus does for us.